Pop culture is full of references to life on other planets. It seems as though humanity is obsessed with the idea of aliens.
There are tons of myths about the origin of aliens and life on other planets, and it's very common to see them used as imagery in different sci-fi movies and novels. And so in this project, we aim to kind of debunk some of these myths and provide some scientific basis to these stories.
Even in more recent example, the movie The Martian shows an attempt to colonize another planet. Our question that we'd like to study is there a possibility for life on other planets, and could we terraform other planets?
Terraforming is manipulating conditions on other planets so that they can become inhabitable to Earth-like standards. Life on Earth is well understood. As we know it, the biosynthesis of materials and substances in the atmosphere of the planet came together to create the essential building blocks of life. Those building blocks became enclosed in membranes and learned how to replicate themselves to survive. Eukaryotes are not the only cells on the planet, though. One of the biggest forms of life on planet Earth are plants. Plants get energy from the sun through the process of photosynthesis. They are able to store sugars. Plants are able to do this by using different pigments to absorb specific wavelengths of light. The major pigments used by photosynthetic organisms on Earth are chlorophyll A and B, which absorb green light.
As light intensity increases, the rate of the light-dependent reaction, therefore photosynthesis, increases proportionally. The more photons of light that fall on a leaf, the greater the number of chlorophyll molecules that are ionized, and more ATP and NADPH are generated. Light-dependent reactions use light energy, and so are not affected by changes in temperature. For our study, we'll focus on two Earth-like bodies, Proxima b and Titan. Titan is the largest moon of Saturn. It is the only moon known to have a dense atmosphere, and only object in space other than Earth where clear evidence of stable bodies of surface liquid have been found. The atmosphere of Titan is largely nitrogen. Minor components lead to the formation of methane and ethane clouds and nitrogen-rich organic smog. The climate, including wind and rain, creates surface features similar to those of Earth, such as dunes, rivers, lakes, seas, and deltas, and is dominated by seasonal weather patterns as on Earth. With its liquids, both surface and subsurface, and robust nitrogen atmosphere, Titan's methane cycle is analogous to Earth's water cycle, at the much lower temperature of about 94 Kelvin.
Proxima b is a small Earth-like body in the system of Proxima Centauri. Proxima Centauri is the closest star to Earth's sun, and it was discovered back in 1915. However, very little is known about this sun and its orbiting bodies. Proxima Centauri is a red dwarf star that's much smaller than Earth's sun. This means that it emits less light, and also that the light is in significantly different wavelengths than that we are used to here on Earth. It's believed that Proxima Centauri could be friendly to life. However, since it's so far away, we can't send a mission there to check and to see if life exists on this planet, and we also can't observe it in high detail.
These are not identical, in which the conditions, specifically temperature, atmosphere, and light, are different. The atmosphere of Earth is composed of methane, hydrogen, ammonia, carbon dioxide, water, and sulfur dioxide. The atmosphere of Proxima b is still unknown to us. The atmosphere of Titan is composed primarily of nitrogen, methane, and ethane. The temperature of early Earth is hotter than the modern day, but we do not know the exact temperature. Proxima b's temperature average is negative 30 degrees Celsius, but is dependent upon their light cycle. The temperature of Titan is colder than Earth. The light in early Earth is a full light spectrum and has a 12-hour light and dark cycle, which is the same as today. Proxima b is a very interesting case because it revolves around a red dwarf star, Proxima Centauri, and has tidal locking, which is when one side of it constantly faces away from the sun and one of it constantly faces the sun. The light side is too hot due to consistently being in the sun and are frequently scorched by solar flares. The dark side is too cold and does not have light for photosynthesis. But there is a band that is just right between the light and the dark, and we have coined this as the Goldilocks zone, which is not too cold, but not too hot, but just right for life. The light on Titan is a full light spectrum and has high UV exposure.
A major limitation to terraforming other planets is that we don't know if plants could evolve to use other wavelengths of light for photosynthesis. Our goal for the first experiment is to determine if plants could evolve to use the light from red dwarf stars like Proxima Centauri. Our hypothesis is that if we shift the light available to photosynthetic bacteria from normal to higher wavelength red light, then the bacteria would evolve to be able to use the red light to generate ATP.
Another major gap in our knowledge is if amino acids and other biomolecules can be synthesized in an atmosphere different from Earth's. For this experiment, we'd like to look at Titan's atmosphere and see if amino acids can be synthesized. So our hypothesis for this experiment is if we recreate the atmosphere of Titan in a sealed container and add energy to mimic storms and meteorite impact in the form of an electric shock, we will see amino acid formation.
In experiment number 1, we want to manipulate the light wavelengths that photosynthetic organisms are exposed to and see if it will have any effects on the yield of ATP. The general design is to grow Synechococcus, a unicellular photosynthetic bacteria, and expose it to different wavelengths and different experimental controls. Expose it to gradually changing wavelengths of light while measuring its ATP yield. The ability to produce ATP with red light exposed wavelengths, it will be our measure of evolutionary progress. The control for the experiment will be bacteria grown in normal white light or the light that we are exposed to on Earth. In order to keep the experiment as close to the conditions found on Proxima b as possible, we won't use the 12-hour light cycle that is here on Earth, but the 24-hour life cycle that is found on Proxima b.
The expected results for the manipulation of light wavelengths to evolve photosynthetic yield of ATP is that over time, as we shift the wavelength, the AT production at goal wavelength should therefore increase. Ideally, we would like to evolve plants to grow on other planets. However, the life cycle of a plant is very long and is much more feasible to use bacteria in a lab. Another limitation is that this study does not take into consideration the amount of light on Proxima b or on Earth. Proxima Centauri is far weaker than our Sun, so plants on Proxima b would receive far less light.
For our second experiment, we will use the methods developed by Miller and Urey in their primordial soup experiment, where they replicated early Earth's atmosphere to show how different biological molecules could be synthesized from lightning strikes. Our general design will have an airtight container with a replicate of Titan's atmosphere inside. We will attach electrodes to the container and send electric shocks through it. We expect to see some amino acids and maybe other biomolecules like nucleotides and sugars form, but we're primarily looking for amino acids. A limitation is that we only account for the atmosphere and liquid components of Titan, but not the solids on its surface.
Significance of our first experiment, changing the wavelength of light to evolve photosynthesis. So the significance of this experiment specifically would be that we would now know if photosynthesis can utilize alternative wavelengths of light to make ATP. If this experiment is successful, future work could be done to identify the mutations that have evolved to adapt photosynthesis to use these alternative wavelengths. And then this mutation could be edited into other genomes to optimize the photosynthetic abilities of different plants and organisms in many different alternative environments. This would also provide us with a basis for which life on other planets could harness alternative wavelengths of light to create photosynthetic energy.
The significance of Experiment 2, the synthetic recreation of a Titan-like primordial soup, would mean that life could exist possibly on other planets. The results to indicate this would be that amino acids would form under the experimental conditions, and that those amino acids could survive under the experimental conditions as well. This would indicate that life could develop on planets that are not identical to Earth in atmospheric composition. Future possible work would be to create other biomolecules from Titan's atmosphere, like ones known on Earth. We could also look at the effects of meteorites would have when they crash in, creating more heat, and possibly pre-existing molecules on Titan already, but that would require flying out there.
And finding life on other planets may not be something that'll happen for hundreds of years, but every step we get closer to understanding life on other planets, but also on Earth, is another step we are to finding more answers. So we may not find little green men, but understanding a little better is going to help us in the end. The sky is the limit. We would like to thank Dr. Regan for all her help on this project, and Dr. Lindner for all his fun space facts and helping us with this. If you have any questions, ask the aliens.